Okay, so I want to start with thanking the organizers for giving me the opportunity to actually show the data at this meeting here. And it looks like it's going to be a really exciting workshop. And maybe he's already doing it. Could we dim the lights a little bit before I start? Yeah. It's also, I think it's nice that I get to start, because then I feel like I can listen to all of your talks very carefully, and I will not be fuzzing about what I have to add to my talk still, or what I have to remove. So I feel like there's a huge advantage of being the first. So it's going to be nice. Okay, the lights are, yes, awesome. Okay, I also have to run back and forth because there's no clicker. So I'm going to be all exercised afterwards. Okay, so I will talk a little bit about some old data that's actually from my postdoc, and then about some really new data that's all not published, and hopefully giving you an idea of what we're interested in and what is the type of data we actually get in our lab. Okay, so just to start, the lab is really interested in how spindles form, right? And most of you know this, but what I find is really interesting that you have all these really small nanoscale components, right? Microtubules, motors, cross-linkers, chromosomes. And during cell division, they kind of magically come together. They form this bipolar spindle, and then that structure can segregate chromosomes into the two arising daughter cells. And what I think is really interesting is that these components are also intrinsically highly dynamic, right? So microtubules in a spindle often have lifetimes of about 10 seconds. Even so, a spindle can persist for many minutes or even hours if you think about xenopus extract. Motors constantly shuttle back and forth, right? And so everything is really highly dynamic intrinsically, yet forms this rather stable bipolar structure. And then somehow all these components together generate forces to first align the chromosomes and then just move them to the daughter cells. And so we really want to understand what are the mechanics of this process, right? So not so much like, let's say, protein A phosphorylates protein B, but really how does that translate into some sort of a mechanical response that will affect how things are moving in those spindles. And so, excuse me. What I think that if you want to understand the mechanics of a spindle, what you need is a much better resolution, right? So this is typical of what you could get from a light microscope, and maybe I just, doesn't work fine. Okay, so this is what you would get from a light microscope, right? But we could all agree that if you were interested in the mechanics, you can't really see the individual building blocks that make the spindle here, right? So what you want is maybe something that looks like this, yeah? Where now you could see every single microtubule that makes a spindle. And so the way we can get this data in the lab is using something that I call large-scale 3D tomography. So I'll walk you briefly through what this actually means and how we do this. And so what we do is we take our sample, and for today, it's going to be C. elegans 1-cell embryos, and it's going to be RPE1 cells. And we do something that's called high-pressure freezing, and this is really just to fix the sample, right? With really good ultrastructural preservation. So we fix it 20,000 kelvins per second, so really fast, and 2,000 bar. And once the sample is frozen, we actually, in a process that we call freeze substitution, just gradually remove all the water. We add fixative, and ultimately we embed the whole thing in plastic. You can see that here at the very end. And once it's in plastic, we can actually section it. So we take the embryos, for example, and just like a sausage, we cut a lot of slices. In our case, those are 200 nanometer thick. And then we collect all these individual slices on these grids, right? Really trying to keep every single one because we want to reconstruct a large structure. So this is how one of these images looks. So this is one section, and in the middle, right, over there in the middle of the picture too, you can see the chromosomes. Spindle poles would be at the sides here, and you can see there's all sorts of stuff around, right? Little red boxes are just the field of view that we get from the electron microscope. The magnification you see here is about 5,000 times, and we have a pixel size of 2.3 nanometers. So it's much higher resolution than you get from the light microscope. And because we want to do three-dimensional structures, we actually have to have a lot of consecutive sections, right? You can see here it says 24. That's for a C. elegans spindle. We've done mouse. That is more like 50 consecutive sections, right? So there's a large number of sections. So once you have this tomogram, of course, now we have to extract some sort of data. And so what we have to do is we have to segment the microtubules because we're interested in those. And when we started, this project, which is like you can see here, more than 10 years ago, we used the Amira software, which is FEI, or now they actually Thermo Fisher. And what this does is it just takes this template cylinder that looks like a microtubule, and then it looks in every voxel of our data if it can find this template cylinder, and then it looks in neighboring voxels to make these microtubules, right? And so this took about 5 hours per tomographic session, just computational time. And the problem is that it actually requires a really significant amount of manual correction, right? So this is really our bottleneck because these sections have thousands of microtubules. And so Amira does a decent job, but it finds a lot of false microtubules. So everything that has a double membrane, it will find for you. And things like, maybe you can see this here. So sometimes there's areas where there's not a lot of contrast. So what happens is that it makes interrupted microtubules, and we have to manually go in and connect those back together. So you can envision that this is quite a laborious task, and it can take days for a single section to actually correct this. So this really, a lot of our time went to sitting in front of a computer and just correcting these traces. More recently, and so you can see it's still a preprint, Robert Kiewicz, together with Tristan Beppler, actually used the machine learning approach, and they call it transformer-based rapid dimensionless instant segmentation, or TARDIS, that uses machine learning to make this process much more reliable and faster. And so what you can see here is tomographic data from plastic, and this is actually cryo-ET data here. You can see the ground truth, which is one of us manually tracing, right, assuming that we do it somewhat all correct. And then you can see TARDIS and Amira, and you can already see on the top one that there's huge differences, right? And you can also see it in the plots next to it. So Amira does a much better job in finding microtubules, in connecting them. And so what this means is that, so not only is the computational time much faster, so one section in TARDIS takes maybe half an hour. Remember, it was like 5 hours in Amira. But it also makes way less error. So the data that comes out is pretty good. And often we can control one section in an afternoon. So you can see that this is really a huge game changer for us, right? And very helpful to speed up data processing. Okay, so to move on, right? Once we have segmented the data, we still have to put it all together in 3D because we want a 3-dimensional volume. And so we use still Amira for this, so there's no other program. Even so, I think this could be improved. And we currently just align, so we first do a coarse alignment just based on vesicles, right, of individual sections. And then Amira uses the model of microtubules to stitch it across D, and then you can get this volume. So you can see here, this one would, for example, be stitched. And then we can also add other features, also using Amira by manually drawing them in. And I think there's now a reverse cloud that kind of allows to train a network to at least segment chromosomes and other features. But you can see that here too, we're still a little limited in how we stitch our data. And I think it would be amazing if we had the possibility to segment everything else, right? Because you can see there's the spindle is here, but there's a lot of other stuff that's around. And I think it would be great if we had a way to segment this and also quantify it all together and how things are related with each other. Okay, so for those that haven't seen, and as I said, that's old data. So this is the data, so you get out of it, right? Now you get a reconstruction. This is a C. elegans metaphase spindle. You can see every microtubule, and you can start being much more quantitative. And it's very pixely today. So these are 20,000 microtubules. You can now annotate in orange kinetochore microtubules that attach to the chromosomes. And you can start trying to get parameters out of this data, right? To understand how these spindles maybe assemble and work mechanically. So what could you do? And I think this is the other thing, right? So when we hit this data, it all looks very nice, but then you look at it and you're like, okay, so what do I do with it? And I'm a cell biologist by training, right? So this was a big challenge, but we'll see. But so the very easy thing, right? You can just take your textbook, you look, how does a spindle look? And you notice that there seems to be subgroups of microtubules, right? There's kinetochore microtubules here in red, and there's astromicrotubules that connect to the cortex. So first step could be you go back to your data and see, can I identify those types of microtubules? And of course, problem is that in tomography, one microtubule just looks like the other, right? So a kinetochore microtubule does not really look different from any other microtubule. So we had to come up with parameters that would define a kinetochore microtubule. And so in this case, we decided it's microtubules that come within a reasonable distance to the surface of the chromosomes. And there's like some ways we determined it, but I'm not going to go into detail. So you can do this, and we chose 250 nanometers. So every microtubule that comes that close to a chromosome could be a kinetochore microtubule. I also want to say that this was relatively easy in C. elegans. We have some mouse data, and I have to admit that the same approach does not work as well. And by eye, it's really hard to determine what actually is a kinetochore microtubule and what not, because they all look the same, and the distance doesn't really apply that much. So not always straightforward. The other thing you may notice is that we actually introduced another group. And so we kind of split the astromicrotubules into a group that goes primarily to the cortex and a group that actually grows towards the bulk of the spindle. And we just do this defined by an opening angle that encloses those chromosomes, right? So every microtubule that goes within this angle towards the chromosomes, we just term spindle microtubules. And then microtubules that go to the cortex, we call astromicrotubules. And again, on the bottom, you can just see the numbers of one example. So it's like 20,000 microtubules, and 500 of those are kinetochore microtubules. So relatively small fraction. But what other properties can you measure, right? And so the next thing you could do is look at the length. And I'm only going to talk about length today, but I want to say that, of course, we can also look at the position of ends. We can look at orientation in respect to the spindle axis. And we also can look at interactions, right? So how close the microtubules come. Do they maybe interact like this? Are they more parallel? But I'm not going to talk about this. So here we just want to show the lengths. And so what you can see, if you look at the length distribution of astromicrotubules, it's exponential. And I think this is what you kind of expect for microtubules, right? They grow and shrink. And the probability for a long microtubule to catastrophe is always higher. So a population will have more short microtubules than long. And you can see it's somewhat like this for the spindle microtubules, not as pronounced as for the astromicrotubules. But what I hope everyone can see is that the kinetochore microtubules look very different, right? So they are not exponential. And if you think about this, and you think about this textbook picture, you can also guess that maybe there's something a little off between the data we have and the textbook picture, right? Because if the textbook picture would be correct, right, where all the microtubules come from the spindle pole, and then some of those become kinetochore microtubules, you would expect a length distribution that hovers around approximately half spindle length, right? I mean, we can agree some maybe might be a little shorter, and those might be a little longer, but overall, it should be much more, it should be kind of flat, and then there should be a peak at half spindle size. And so what this told us that actually most of these kinetochore microtubules are not attaching to the spindle poles in C. elegans, right? And another way to look at it is just looking at the end distribution. And you see this here. So if you plot where the ends are for the different groups, you can see that for kinetochore microtubules, so this is the centrosome at 0 along the spindle axis, you can see that there's ends everywhere along the spindle axis. But then if you look at the spindle microtubules, you can see that the majority of ends actually is very close to the centrosome, right? And again, saying that in reality, this cartoon is somewhat not correct, and it looks maybe a little bit more like this naively, right? So most of the kinetochore microtubules here in C. elegans are actually not directly connecting to the pole. Okay, so now we're going to move to more new data. So this is actually data from Thomas' lab, right? So this is a HeLa cell now. And I'm showing this to make the point that what we found for C. elegans is actually also true in the HeLa cells, okay? And so you can see this here on the plot. So half of the kinetochore microtubules, you see this here, are also not coming very close to the pole. And you can see this up on top here too, where the black dots are the ends. Just ignore these points. The figure was bigger. I just removed parts of it, and I could not erase within the spindle without erasing the microtubules. But so what this shows, right, is that there's ends everywhere too in tissue culture cells, right? Suggesting that really it's a more general thing that kinetochore microtubules are not directly connecting to the spindle pole. We'll stick with this cell for a little bit. The other thing I think that's interesting is that if you look at the length distribution of those microtubules, we can see that the average length of a microtubule is about 2 micrometers. And so that tells us that the majority of those microtubules actually does not span the entire spindle length, right? If you think about the cartoon, I think it's here, right? Microtubules are long and continuous. They all come from the pole, and they span the entire distance of the spindle, so that is not true, right? If you look at this spindle, microtubules are way shorter. And actually, these are just examples of other spindles we've done. So you see some C. elegans meiotic spindles on top, the C. elegans embryo on the bottom, and half of a mouse spindle. It's cut at the centrosomes. But all of those microtubules actually have a lot of short microtubules, and the average microtubule spans just 20% of the spindle. And so that means that spindles are not made of long continuous microtubules, very likely, but just kind of shorter microtubules that, I don't know, some array of shorter, maybe overlapping microtubules, right? Okay, so if we take a closer look, what we can also see is that if you look at the microtubules in the spindle, right, and now thinking about those short ones, then we have the kinetochore microtubules and the centriola, so the pole microtubules, and this is just those microtubules in the reconstructions, the number along the spindle lengths, and then we have those other microtubules. I'll just call them other because they don't really have a name here. And you can see that there's actually a lot of them, right? So about half of the microtubules that you can see in these spindles are not assigned to like the classic group in the sense of that they're astromicrotubules or centrosomal microtubules or kinetochore microtubules. They're just others. And so we were really interested in what are those other microtubules, right? And so, of course, you could say like, okay, so we all know there's bulk nucleation of microtubules, right? In the spindle, there's augment that drives nucleation of microtubules of other microtubules, yeah? And so the first thing we thought is, okay, could we, and this is a little complex to explain, could we look at these microtubules, assign a polarity to them, and then figure out where those microtubules come from and what they are, okay? And so I'm going to walk you through. So if this is a spindle, right, then we take all these short microtubules, we take the end that is closest to the nearest pole. So for example, one end is here and one is there. Then we take this end, assign it to this pole, okay? And then the plus end would be this direction, okay? This would be the minus end closest to the pole and the plus end here. And you can do this for all microtubules, maybe thinking a little bit like augment, right? You have a microtubule, another one coming off. So they should all have polarity determined by the nearest pole. Or alternatively, maybe it's random, right? You just assign random polarity to all those microtubules. So now you need a way to test this. And one way you could go about it is to use second harmonic generation microscopy, right? And so this is what Dan has actually developed in his lab, and I'm pretty sure that's data from your lab too. So I hope you don't have it on your slide. Okay, but you can take a 2-photon image, right, of the tubulin in the spindle, and then you take the second harmonic generation image that allows you to determine the polarity. So if 2 microtubules are parallel, the signal will amplify, and you should see a signal. If they're antiparallel, the signal cancels out, and it will be dark. And so you can use this together with the tubulin density to develop a polarity profile. So how does this look? The black line is what you get out of the second harmonic image. Okay, and it's along the spindle axis. So pole here, and the second pole is at 1. If you now take the model where you just took the end that's closest to a pole and assign polarity in this way, that is the polarity profile you get, the blue line, for everyone that looks over there. And that looks very different, right? So it kind of doesn't match that much. If you take, and maybe I'll just go over here for now. So if you look at the random assigned polarity, so this kind of looks much better, right? We could agree that this seems to fit the data much better. But of course, this is bulk measurements, and we don't really know. So we thought maybe there's a better way to look at it, right? And so everything I showed you so far is using this serial section approach, right? Where we have all these sections, we stitch it all together, we get this really large reconstructions. So good is we have a full structure, and we have all the cellular context. Bad is resolution is maybe not high enough to look in more detail at those microtubules. So how about doing cryo-electron tomography, right? So now you can see those are microtubules. You can already see you have a much better resolution. So advantage is you have this really high resolution. Disadvantage is that in reality, if you do this in a cell, that is the area you're going to be looking at, right? So you lose your entire cellular context based on how you have to prepare those samples. And so I'll just briefly show you in the next slide, right? And so here I have to give a huge shout out to Will Conway, a postdoc at the New York Center for Structural Biology, and he has done all of that work. And I just want to say it's really not easy for anyone that has tried to do any cryo-ET in a cell. It's a real pain. And it took quite some time, but you'll see. So he really nailed it. And so here's Will again, seeing him doing the experiment. And so it's a good one, no? And so what we have to do is we have to synchronize RPE1 cells to enrich for mitotic cells. And you can do this just by a shake-off. Then you put these cells on a grid and you plunge freeze now. And you can see those are the cells. Here's a zoom in. We have markers for tubulin and the kinetochore. So we can really pick a cell that's in metaphase. And then the thing is, because a cell is too thick for the electron beam to penetrate through, you have to cut everything away but 200 nanometer thin lamella. So this is the cartoon for it. And this is how it looks, right? You can see the look, the grid looks also very beaten up, I think, but there's all sorts of lamellas here. And this is how the lamella looks from the side, right? So we can agree that the cell probably was kind of all around here. And now we are left with this really thin stripe. And so you can also guess that if you have a small structure of interest you're interested in, targeting is a huge problem, right? Finding what you want to look for is not easy. Okay, this is just a different picture to while the lamella is very thin to make the point that actually in X and Y, it's not such a small area. And again, here's the grid. Here's the lamella from the top, right? And here is a lamella where we have fitted in some of the tomograms, right? And this also allows us to kind of know our orientation at least a little bit, right? So we would know, are we close to the pole, closer to the chromosomes, kind of what area are we looking in? Because these fields of use are much, much smaller than anything we've looked at before. So this is how it looks. And there's a lot of data on the site here. If you're interested in acquisition and processing, let me start this. This is a tomogram for everyone that has never seen one. It probably looks all gray and gray. People that have seen one maybe already spotted some microtubules. If not, I'll make it a little easier. So you will see microtubules not coming in eventually here. There's some microtubules. It's like very glitchy somehow. There's a delay. There's ribosomes here. And there's actually all sorts of membrane too. I don't know, maybe ER, maybe left over of the nuclear envelope. And here you can see, this is how it looks. Well, okay, there's a delay here. I see different things here than you. Okay, so this is how it would look, right? So you can see now there's this reconstruction of microtubules. So the question is, what can we do with this data that we could not do in plastic, right? And so while I had high hopes that maybe we're going to see a motor or whatever else, spoiler, no motors here. But there was one parameter we could get, and you'll see it's actually super interesting. So the first thing you could look at is the spacing. And you can see this here. So this is just the nearest neighbor of microtubules. And you can see that their microtubules in this homograms are arranged with a 33 nanometer spacing. And then the exciting thing that we could get out is actually the polarity of individual microtubules. So that's something we could not see, right, in the plastic. We had to make some weird guesses on what the polarity would be. And so if you look here, you can see that they are antiparallel. And you can also see in my title saying, metaphase microtubules are organized in interdigitated and antiparallel bundles with 33 nanometer mean spacing.
Sorry, I may have missed this, but can you just comment on how you identify the polarity?
Yeah, you can do subtomogram averaging, and if you look at the cross section of the microtubule, you can see a slew, for the protofilaments. You can see I had the picture in. Maybe I will pull it up, but you can basically it's like little hooks that point one way or the other. And so this is a long title, and what this actually means is that if you look at the spindle, and I apologize for everyone that's colorblind because it's red and green, I know. But let me tell you that what we see is that there's antiparallel, local antiparallel microtubules in those spindles, right? So you see some in green, some in red. And so, okay, that was kind of interesting. And we thought we follow this up a little bit more. And so one explanation is that maybe those are just because of bipolarity, right? So naively, you could think if you have a spindle and you have some microtubules coming over from the other side, those are somewhat antiparallel, and maybe that's what we're looking at. And so one way to test it is you can treat the cells with centrinone, so that prevents duplication of the spindle pole, and you end up with a monopolar spindle, right? Somewhat like this. And so if we do this, you can see that the spacing was really not affected in those spindles, still the same. And you also see, you see it here or here, that there's still antiparallel microtubules. Okay, so that kind of suggests that these antiparallel microtubules are not, so bipolarity is not a prerequisite to have these antiparallel microtubules. And naively, in a cartoon, it would be somewhat like this, right? You still have a monopolar spindle, and there's these antiparallel microtubules in this spindle. So that was surprising. And so now we have to walk a little bit back. So if we think about spindle assembly, right, then it's not only the microtubules, but it's the interaction of microtubules and motors that drives the assembly, right? So you have microtubules, you have motors. Sometimes they just walk on a microtubule, but most of the times they can cross-link them and they can slide them in respect to each other, right? And all of these interactions ultimately give you the bipolar spindle. Now, if we talk about two of the maybe best described motors, I don't know, we're going to talk about kinesin 5 or EC5, right, which is a plus end directed kinesin. It can drive antiparallel sliding of microtubules. And we're going to talk about dynein, which is minus end directed and does clustering of microtubules. And so in a spindle, you inhibit kinesin 5, EC5, you end up with a monopolar spindle, right? You can see this here. Inhibition of dynein causes this disorganized spindles, okay, where microtubules are kind of, you can see there's no poles really, and everything is somewhat disorganized. Interestingly, and this is data from Sophie Demont's lab, if you take cells that are depleted of dynein, you can see this here, they're disorganized, and you inhibit kinesin 5, these spindles can actually restore bipolarity, okay? So suggesting that it's the balance of plus and minus end directed motors that helps to establish bipolarity of spindles. Okay, so we decided, okay, well, look what happens to microtubules in this setup. And so the first thing you can do is you can inhibit kinesin 5 with monostrol or STLC, and that causes monopolar spindles. And I'm putting in quotation marks because it causes spindles that have two collapsed poles, right? So there would be still two centrosomes, but they collapse into one bigger pole, and so you end up with monopolar spindles. There's a lot of walking here. And so I think you can see this immediately. So if we look at monostrol-treated spindles, you can see that most of the microtubules in these monopolar spindles are actually now parallel, right? You see this here. There's one, wrong one maybe here. And you can see this in my cartoon. Okay, so suggesting that somehow maybe kinesin 5 is what drives these antiparallel bundles in the spindle. If you look at dynein, you can see dynein is here. So dynein did nothing to the polarity of the microtubules. And this is just another way to look at it, right? This is control. This is a EC5 active monopolar spindle with centrinone, still monopolar spindle with monostrol, parallel. And here's the dynein inhibited spindle. And there you can see that it's, again, antiparallel. And now I have stuck new slides in here this morning. So maybe from here on it will get a little bumpy, but we'll see. Okay, so the other thing that we see is that if we deplete dynein, we can see changes in the spacing of microtubules. And maybe that kind of makes sense if you think that this spindle is really disorganized. And if you look at the spindle here, right? So this is the 33 nanometers, this is dynein, and then you can see all these peaks, right? Showing that dynein really affects the spacing of the microtubule in the spindle. And again, this is just another way to look at it, right? This is the spacing in all the conditions here, the bipolar and monopolar, EC5 active or inactive, kind of never really affected. And then here in dynein, we get this disorganized spindle, and we affect the spacing between microtubules. Okay. And so now I told you before that if you get rid of both motors, you can restore bipolarity. So obviously, we knocked out both motors and looked at the cryotomography data. And so one thing you can see is that now this is what happens if you get rid of both. Okay, and you can see that you kind of restore the spacing actually, right? It is no longer that disorganized. It actually kind of falls back to where it was before. And you can see this here too. Okay, so really suggesting that dynein regulates the spacing between the microtubules. And I just want to show this. So this is, again, the same plot. This is one of the spindles, and this is the spacing along the spindle axis. Okay, and you can see that it varies. But if you plot the spacing against the density, you can see that the spacing of the microtubules actually varies with the density. So suggesting that dynein might regulate the spacing between the microtubules by actually regulating the local density, right? So it kind of clusters microtubules, and in this way, it can regulate the density of microtubules in spindles. So now comes the more interesting part. The other thing is that if we look at the polarity of microtubules in the spindles that are co-depleted of kinesin and dynein, we can actually see that the microtubules here become way more parallel, right? So I think I have a cartoon.
One question. The arrangements of microtubules, is there any kind of crystalline ordering in angular direction, or is it more like randomized?
We didn't look at the angle, but maybe that's a thing we should look at. We had not looked, yeah. I mean, we can do this in a plastic data, I think. We kind of, but yeah. Okay, so what this means is that this bipolar spindle that has neither kinesin nor an active EC5 is somewhat composed of parallel microtubules, right? So this is what we can see in the tomography data. So if you inhibit kinesin 5 in the spindle, microtubules are more parallel. And I want to show one thing. So this is again from Sophie Redeman's group. And what they do here is they have this little setup where they can squish spindles, right? You can see that here. And this is a control. And I mean, not so much is happening apart from that the cell is like crazy blabbing because you squish it. See at the bottom here, you can actually see that these spindles kind of disintegrate, right? They seem to fall apart. And so what we're thinking is that, so is this actually because those spindles are mainly composed of parallel microtubules instead of antiparallel microtubules. And if a spindle has more parallel microtubules, you lose a lot of the cross-linking ability. And that could actually affect the stability of a spindle and explain why those spindles are mechanically much more fragile. Okay, and so this is a little cartoon to kind of sum up what I showed you. And so I just want to point out, right? So we think kinesin 5 has extensile forces, so it kind of slides the antiparallel microtubules. Dynein has more contractile forces. It will cluster the minus ends. And so if you look at this control metaphase spindle, you can see that it has all these antiparallel bundles. If you inhibit the kinesin 5, you end up with more parallel bundles at a monopole. If you inhibit dynein, you end up with antiparallel bundles, but spacing is kind of mixed up. And maybe this is because you have excessive extensile forces now. If on each of the cases, you inhibit here dynein or kinesin, you can restore bipolarity and you end up with parallel bundles, right? And so I think this, this observation is actually really important in the terms of how we think about spindles, right? Because I think if you want to come up with a model of how a spindle mechanically works or assembles, it kind of matters on what is the orientation of your microtubules, right? Because you're going to get it all wrong if they all point in the wrong way. So I think understanding that there is a higher fraction of antiparallel microtubules is actually highly relevant in how we think about spindles. And so I put one last slide because there's nothing about machine learning in my presentation. But still, I think so. I think there's a lot of room for how it could be super helpful for us. And so I kind of talked about this before, right? Segmentation, because there is so much more in spindles than microtubules, and I think there's all this membrane and all sorts of other stuff. And it would be really cool if we had a good way to segment it and also really quantify it. How do sometimes microtubules come close to membranes, and I mean, you could envision that they're doing something and it has some consequences, right? So it would be really cool if we could see this. Stitching in 3D still a bit tricky and kind of hard to do. Data analysis and interpretation is also a big thing because there's so much data that sometimes it's really hard to figure out how you retrieve anything that could be used and important. And I think maybe some sort of predictions, because we do often large scale volumes, but that is very laborious. So if we had a way to have smaller volumes, or from the cryo-ET, right, you have this tiny volume, but if there's a good way to have these small volumes and then still think about how a larger volume could look in terms of making some predictions, I think that would be helpful too. Not sure machine learning can do this, but I think those are things that would be really amazing if we were able to do any of those. And with this, I want to thank every one of you, the organizers, our funding sources, everyone in the lab, all the collaborators, and Will. And I'm happy for any question.
We have time for like 2-3 questions. Yeah.
2-3 questions, but I'm going to ask really fast.
I'll also be here.
What?
I'll be here until Thursday.
Yeah. No, you said, so the 3, you can answer them all. They're just technical. This was really lovely, Stefanie. It totally made me think about a lot of things. But you said you saw no motors in that EM, so I was curious about that. Also, you guys, the spindle field, I assume, has done these sorts of high resolution EMs before, collected this sort of data in spindles or not. So I was curious whether there's other data out there that was already revealing some of the sort of packing and alignment, and that could have been done, or whether it's really that we have to create these data sets each time. And then I'll do the last one in a second.
Okay. So we could not see motors, and I think it's because many things. The signal to noise ratio is really crappy in cytoplasm. So if you do this in vitro, I'm sure you can see motors. In vivo, you just fight with signal to noise. And also the problem is in vitro, you know which motor you put. So resolution comes with ability to average. If I don't know where is which motor, I can't average, and I'll just see nothing. So if you need it to be, I think there's ways to make it work in the future, but right now we couldn't see.
And then the packing, when you did the double knockout, was smaller in your peak.
It had 2 peaks, yeah. And then there is, Dick McIntosh did some larger scale reconstructions, but they are done in different ways. But I think you could go back, and then those are the first ones that did it in a new way. And I think because it is very labor intensive, not a lot of people do it. And so what people often do is smaller areas, right? But you could get some packaging, I think, from this.
Just curious about how to bring all the data that's out there together.
Also, the packaging, if you do plastic, there is deformation, right? So you don't know if what you're looking at, and it's anisotropic. So not every region of the embryo will shrink in the same way. So you kind of don't know if you can trust it or not.
I haven't thought about this for 15 years. I apologize. My question might be super stupid, and maybe also more for Dan. But if you have this centrinone thing, you make a monopole which would now have actually antiparallel flux. Has anyone done that experiment?
Nope.
And you do it?
Maybe then.
Yeah.
Okay, but that would be a clear prediction.
Yes, yes.
Okay.
Yeah, from this, there's a lot of things you could, right?
And really get that polarity through dynamics instead of...
Totally.
Go ahead.
I also have a question about the antiparallel microtubules. So how do you think they're nucleated in different orientations, or do you think smaller microtubules are made by severing and then somehow reoriented by the...
See, Radhika got the thing I removed from my slide because I did have a slide with open questions, and it's exactly this. So I have no idea, right? So, and either they're transported there, who knows, right? But maybe they're also, I don't know, at the end, if you think about augment, maybe if a stub is small enough, or maybe they detach and they can be, maybe they detach if the mother microtubule falls apart and then they flip around and they get, I have no clue. But this is definitely something one can look into too, right? There's ways to test what is required. So yeah, we don't know.
Okay, one last question.
Sorry. I just got a question. So have people who track EB1 or EB3 comets, actually it should be me, but anyway, we also track what we've never seen so much, have others who track seen in both ways? Growth
That is
in
a very
both-
good
I
question,
don't-
right? And so we're talking about
I've never looked at it.
yeah,
Throw away all that data as noise.
yeah, I don't know. So I mean, it's also inside spindles, right? Which is far denser than anywhere. I think maybe. So I know in C. elegans, people have reported that occasionally there's a microtubule going the other way.
Yeah.
You also don't know if they're growing. I mean, there's a lot of assumptions you would have to make, right? But I think so too. I mean, you could test some of these things too by some smart speckles or fraps or anything, right? So it's not untestable.